So the next speaker will be Yakov Tsipilov, and he will be talking about loci and genes involved in chronic musculoskeletal pain identified by analysis of genetically orthogonal pain phenotypes. So the floor is yours.
Okay, thank you very much. Can you hear me?
Yes, we can hear you.
Okay,
Just please
so,
full screen.
yep. Hello everyone again. Today I want to talk about our study of the genetics of the pain. Musculoskeletal pain and disorders are actually one of the most common conditions. Among the most prevalent and the most famous one are back pain, neck pain, knee pain, and another pains in another sides of the body. Uh, uh, Musculoskeletal pain affects at least 1/3 of all adults, and in up to 50% of the cases, it develops the chronic state that is currently is top 10 cause of absenteeism and costs to the European countries quite a lot. These conditions are actually a group of mixed pain syndromes with different pathologies and pathophysiology. And given its very heterogeneity base, it's quite complicated to study. Among the known risk factors are female gender, some social factors that includes depression and anxiety, unhealthy lifestyle that includes smoking and obesity, and genetics. One of the dominant models of the pain conceptualization is so-called biopsychosocial model that says that the pain is the consequence of several factors. One of them biological or morphological factors, like something that really triggers the pain, some problems in cartilage, diseases or inflammation. And another factors relates to social status, cultural factors, the current mood, and how a person actually percept the pain. So up to now, quite a few GWASs were conducted on different site-specific pain. In general, musculoskeletal pain has quite big heritability, up to 60%. And already few genes were reported to be associated with different types of the pain. The main problem studying the pain phenotype is that you need to have a huge sample size. Here I depicted, Here I depicted the Manhattan plot from our recent back pain study, where we use UK Biobank with more than 300,000 people, and we discovered and replicated only 3 loci. So again, huge sample sizes are needed to do the proper genetic study, or probably we need some new methodology to deal with heterogeneity. It was observed before that different musculoskeletal pain actually have some common genetic background. And obviously, if we somehow could extract this shared genetic background between pains, we could possibly reduce the heterogeneity in these phenotypes and increase the power of our analysis. So the aim of our study was to extract this shared background and overcome the heterogeneity. To do it, we use the method of genetically independent phenotypes. The idea is very simple. If the group of variables has something in common and expected variance explained by this factor is big, you can use the principal component analysis. For this, usually matrices of the phenotypic covariances is used. but and eigenvalues of the this matrix of the correlations is usually used as the coefficients of linear combinations or so-called loadings. In our case, we use not the matrix of phenotypic covariances, but the genetic covariances. And it should be noted that if you have GWASs for regional traits, you can obtain GWAS for any linear combination of regional traits using quite simple math. So if you have loadings that came from the matrix of the genetic covariance, you can obtain the GWASs for each of the component that will be genetically independent by definition. Some interesting and beneficial properties of this method that actually you can use it only in case of only using the summary statistics. So you can obtain the genetic covariance matrix using canonical software, for example, LD-score regression. And the phenotypic correlation matrix can be obtained using the correlation of Z-scores. Also, one of the interesting properties is actually it can work with overlapping and non-overlapping samples. It can combine logistic and linear regression. And also, in contrary with some multivariate approaches like MANOVA, which obtains different linear combination of the traits for different SNPs, it has only one linear combination for each SNP, so it can be used in different in silico follow-ups, like for regular GWASs, that includes Mendel randomization, colocalization methods, and genetic correlations. Among the limitations of this method, we have that it works only if you have not less than 3 traits. So here I depicted the scheme of our study. So we used 4 musculoskeletal pains and UK Biobank that was split by discovery and replication cohorts. We obtained the matrix of genetic covariances and loadings that were used to construct the genetically independent phenotype GWASs, and then these components was used to construct the replication GWASs as well. We performed the replication of our findings and did a variety of in silico follow-up techniques that includes gene set enrichment, colocalization with gene expression and complex traits, genetic correlations, fine mapping, and variant effect prediction. So here are results of the method of the construction of genetically independent phenotypes. In the first figure, the coefficients, the loadings of the linear coefficient, the linear combination are depicted. So for the first component, we can see that it looks like the sum of the panes. Basically, it has sense just because the comorbidity of the pain phenotype not so huge. This method actually depicted the property of this method that we actually can sum all our pains and we will obtain the phenotypes that will depict what they have in common. So in this figure, we depicted the genetic variants explained in different pain phenotypes by the principal components. The blue color corresponds to the first principal component, and as you can see, it presents in all pain phenotypes. And another color that corresponds to another component presents only in 3 phenotypes. So the first component is by definition is something that they share by common, and we can call it shared heredity. So another interesting interesting observation is that the heritability of the first principal component was twice as higher than the heritability of the most prevalent pain phenotype. So it was, so the most heritable trait was back pain. This heritability on observed scale 4%, and the heritability of the first component was 7%. So here is the GWAS for the first component is presented. We discovered and replicated 5 loci and decisively prioritized 3 genes. Among other 3 candidates, we also has the list of genes, but among the most interesting observations were the gene set enrichment that depicted that we have quite a lot of terms, significant terms related to nervous system. Also, if we recalculate the SNP-based heritability using Gremel method and convert it to the liability scale, we discover that the first component has 27% on liability scale. That is quite impressive. We also performed the genetic correlation in this slide. The matrix of the genetic correlation between first principal component and all its genetically correlated traits is presented. So it can be seen that all correlated traits form 3 or 4 genetic clusters. One of them related to obesity, another one related to depression and mood and other mood traits. And the biggest cluster is related to social demographic traits that includes cancer, age and death of the parents, education, and another demographic factors. Interestingly to see that first principal component actually mostly correlates with this social demographic cluster. It should be noted that also we performed the analysis of another components, but we found only one additional gene for the second component that was GDF5, the known gene that is associated with osteoarthritis. And nothing conclusive can be given based on the results. In summary, based on the results of gene set enrichment found genes, genetic correlations, we concluded that the first principal component actually represents this nerve or biopsychological component of the pain. And most probably it it corresponds to pain perception and processing. And all our results basically support the biopsychosocial model of the pain. So in general, they present quite simple method that can be used to extract some shared genetic background between different traits. They found 6 new significant and replicated loci associated with musculoskeletal pain, and we prioritized few genes that further can be used in vitro follow-ups. I think that's it. The publication is coming soon, but all details and links to the software can be found in our paper that put it in bioRxiv.
Thank you very much. We have time for some questions. I don't see a question at the moment. I would have some myself. Maybe I missed something. So when you extract the principal components from your genotypes, you would normally use them for correction for population stratification. So maybe you can comment on this a bit. How the relation.
Actually, the idea of this method is not how we can correct the principle, how we can correct the population stratification. So this method is applied not on the kinship matrix. This method is applied to the matrix of the genetic correlations between traits.
Yeah.
So this is not something that's called genetical principal components. It's it's something that relates to the traits, not to the population stratification.
Okay, thank you. There's an anonymous question. How would your design have worked if you had just summed the sites of pain? I assume used SNPs that were shown to associate with pain rather than making your principal components.
Didn't get the question. Could you please repeat?
Yeah, I have also some difficulty. So as I understand it is, if you had started with SNPs that are shown to be associated with pain and worked with those rather than taking your whole principal components, would this have made a difference?
I'm not sure that I did this question correctly, but I think it relates to the something that called, let's use the pleiotropic SNPs only and study the SNPs that were associated with all these traits at the same time. So basically, yes, we could construct somehow the list of SNPs that were pleiotropic with all of these original traits at the same time. But in that case, we could not study this component using the huge variety of the techniques like genetic correlation, Mendelian randomization, and colocalization methods. So yeah, I hope it answers your question.
Yeah. Thank you. I think otherwise you will be available in the session after that. A final question from Nicola. Do you think that pain associates with socioeconomic factors due to type of work?
Actually, it is. So the type of work, one of the risk factors for some of the site-specific pain. For example, heavy work is one of the factors for back pain. The question is how this, the genetic component of the predisposition to heavy work is reflects in the GWAS of back pain. This is another question. So we actually now trying somehow to decompose the genetic component of the back pain and see whether it's just the construct of all its risk factors, or we have some unique component that really reflect something related to back pain.
Yeah, thank you very much. Again, let's raise hands for Yakov. We have to move on. The next speaker will be Kaur Alaso, and he will talk about matrix factorization reveals cell-specific trans-acting regulatory variants controlling modules of co-expressed genes.
I'm waiting to be able to share the screen.
Please try again.
Okay.
Okay, the floor is yours.
And I'll add video as well. Can you see my video now?
So everything is fine. If you full screen your presentation, you're good to
go. Okay. I changed this title from matrix factorization to co-expression analysis because the methods are a bit more general than just matrix factorization. But to start, I think the main aim of human genetics, like one of the main aims of human genetics, is understanding what are the causal factors or processes that lead to complex traits, complex diseases, and how can these be kind of then modified, either pharmacologically or socially. So the kind of the first step to reaching this goal is GWAS. But GWAS doesn't really identify mechanisms. GWAS just finds genetic variants that are associated with the trait. So if you want to get closer to the mechanism, we can turn to things like eQTLs, which can then link genetic variants, disease-associated variants, to some genes that these variants influence. And if these cis eQTLs are cell type specific, then they can also identify the disease-relevant context. So we might find out that this particular variant influenced the expression of this gene in monocytes, while this one influenced the expression of this gene in liver. But still, we just got a list of genes. We still don't know anything about the mechanism. What we really want to achieve is we want to find what are the kind of the cellular and molecular processes in between, from genes to the disease. And in case of coronary artery disease and FH, that's one of the few cases where we notice that one of these risk factors, these molecular processes, is cholesterol metabolism. And if we have that, we can use these disease-associated variants that act through these genes as instruments in Mendelian randomization to assess causality. And this works beautifully for cardiovascular coronary artery disease and not so beautifully in other cases. So what I'm going to try to, but what if we have not measured these intermediate molecular processes? And this is, I believe this is the case for most complex diseases, that we actually don't know what these processes are and we haven't measured them. So what I'm going to try to convince you today is that sometimes we can use co-expression analysis, gene co-expression analysis, to infer these molecular processes from expression data. This is not a novel idea. There's a nice paper already from 2011 from Maxime Rotival and colleagues, where they studied this X, which is a gene expression matrix from monocytes. They had a few hundred individuals, and then there's like 20,000 genes over here. And then they used independent component analysis to decompose this large gene expression data sets into independent components and their activity patterns across individuals. So instead of doing a GWAS or a trans eQTL analysis for 20,000 genes, you can only use like few tens or maybe 100 independent phenotypes that have been extracted from the original data. So there are many different ways to identify these co-expression models from gene expression data. So you can start from one kind of class of methods are these matrix factorization methods, like ICA, but also PEER, that try to decompose, try to find components in the data that maximally explain the variance in the data. So you can try to decompose the full data matrix into independent components. And then there's kind of another class of methods which are more like bottom-up methods, that they try to identify groups of genes that have highly correlated gene expression levels. One of these is WGCNA. And then kind of more recent additions to these are two methods in these classes that incorporate prior information about regulatory interactions, so either kind of known pathways or gene ontology terms and so on. And for factor analysis, this is called PLIER, and for gene co-expression analysis, there's a method called FuncExplorer. And so while ICA, WGCNA, and PLIER have previously been used for trans eQTL analysis, this has been done in only select cell types, so basically just monocytes, and only in whole blood. So this work here that I'm going to talk about today has been led by Liis, who's a PhD student at the University of Tartu. So what Liis did is that she first collected large gene expression genotype data from 6 blood cell types and 3 stimulated conditions from 5 different published studies. The cis-eqtls are all available with the eqtl catalog from these studies. So the sample size varies from 220 to up to 700 in monocytes. And then what Lise did next was she took the gene expression data and then used these 5 co-expression analysis methods to identify modules of co-expressed genes. But the key thing was here that she did it on the full data set, so where all of the 9 cell types and conditions were together, analyzed together, but she also did it separately in each cell type and condition. And kind of the justification being that there might be highly cell type specific like genes that are co-expressed in only one cell type. And if you analyze them together, you might miss these gene modules. And then finally, she did trans-eqtl analysis in the end for these, using these gene expression modules as traits. So these approaches identified between 50 and 150 independent modules in these different cell types and tissues, either when we analyze them together or separately. And the nice thing is that when we do this trans-eqtl analysis, this plot now shows 303 nominal significant associations, so P less than 5 × 10⁻⁸, but this doesn't account for the number of modules we tested or the number of methods and cell types. So these are just nominal significant associations, but the nice thing to see here is that the 2 of the strongest hits have been replicated in multiple independent data sets beforehand, and actually Helene just showed these 2 loci, the lysozyme and interferon beta, in her slides as well. One of the other ones that I'm highlighting here, ARCF3, has been previously found in, I think, 2 or 3 different whole blood trans-eqtl data sets. And we have one completely novel association as well. And so I'll now focus on these 2 examples to kind of show what we can find from these data sets. So ARCF3 locus. So what we see is we detect the trans-eqtl for a cluster, for a module of co-expressed genes in platelets. This is the lead variant. It's also a cis-eqtl for ARCF3 gene, and it very clearly co-localizes with a signal for mean platelet volume in GWAS. And this is, like, the signal is extremely strong. When we look at the lead trans-eqtl variant, we find that it's associated with 3 co-expression modules in platelets, and basically there's no association in any of the other cell types. So this is highly platelet-specific association that we find. And when we look at the module genes, so here's the cis-eqtl for ARCF3, and these are all of the genes that are associated in trans with this variant. And when we do enrichment analysis, we find that this group of genes is very strongly enriched for platelet-specific functions. So they're basically very strongly enriched for terms related to platelet activation, which I think makes sense in the context that this is also a GWAS hit for platelet-related traits of mean platelet volume. The other association that we find is at the SLC39A8 locus. So what we see here is we have a trans-eqtl on chromosome 4 in monocyte stimulated LPS for 24 hours. Interestingly, there's no corresponding cis-eqtl in our data set. So when we look at the naive monocytes, there is a cis-eqtl, but it's an independent signal. And the trans-eqtl lead variant is not a cis-eqtl lead variant here. So we were kind of puzzled by this for a long time, but then we found another additional eqtl data set from Kim Melmoth et al. And basically what we discovered is that this trans-eqtl lead variant is a transient cis-eqtl for this SLC39A8 gene. So we see a cis-eqtl, we don't see cis-eqtl in naive cells. We see an effect 90 minutes after stimulation, and then the cis effect disappears already by 6 hours. But then the trans effect that we see is present still at 24 hours after stimulation. This trans eQTL was detected by only one co-expression cluster, only in stimulated monocytes. So we don't see this in any other condition or cell type. And interestingly, it's associated, there are 7 target genes, but they're all in the same locus on chromosome 16. And when we look at what these genes are, they are metallothionein genes, so they're very strongly enriched for zinc ion homeostasis and zinc ion binding. But they're also enriched for a binding motif of a metal transcription factor 1, MTF1 transcription factor motif. So we also see from our data that SLC39A8 is very strongly upregulated in response to LPS. So this is log scale, so it's log 2 fold change is 3.5, so it's about 10-fold upregulated in response to stimulation. And so here's kind of a cartoon of what we think is going on. There's lots of literature evidence for these multiple steps, but what we think is that LPS stimulation activates signaling pathways, just like NF-kappa signaling, but there also might be other ones involved. And then in response to LPS, SLC39A8 is upregulated, as you saw here. And there's this cis-eQTL effect that we see in this immediate early upregulation of SLC39A8. This leads to increased accumulation of the CYP8 protein, which is the protein encoded by the SLC39A8. And then in LPS response, CYP8 is responsible for importing zinc ions into the intracellular space. And then MTF1 transcription factor is activated by zinc ions. When the zinc ion concentration increases, MTF1 is activated, and then MTF1 binds to these trans eQTL genes that we detect in this co-expression cluster. They are known targets of this MTF1 transcription factor. So that's kind of the model that we have in our mind here. So to conclude, we think that co-expression analysis can really identify interpretable trans eQTLs, especially when we combine it with this gene ontology and pathway enrichment analysis. We can in some cases, we can really interpret what the trans eQTL seems to be doing. And it can also reduce the number of tests performed, because instead of 20,000 phenotypes, we're only testing a few hundred phenotypes. But there are caveats to this approach. The power now also depends on the power to detect associations. Now also depends on the power to detect co-expression clusters. So if we use a method that misses this particular co-expression cluster, we also won't find a trans eQTL for that. And then obviously we have sample size limitations that trans eQTL is still quite hard to find because the effects are small. But we believe that if we aggregate across multiple co-expression methods that optimize for different aspects of these co-expression modules, so top-down optimizing for maximizing variance explained in the dataset versus bottom-up co-expression clustering, looking for really kind of small groups of genes that are highly correlated in expression, this can really kind of overcome some of the method-specific biases. But really a key open question is should we be performing this aggregation at the level of gene expression like Liis has done here, or should we be doing it at the level of genetic association, like in the Atlas QTL method that Helen presented earlier today? And with that, I'd like to thank Liis, who's been doing most of the work in this project, and also Nurlan and Hedi, who have contributed. And we're looking for a postdoc who would continue this work on much larger RNA-seq data sets that we've collected for the eQTL catalog. So we have thousands of RNA-seq samples where we would like to keep doing trans eQTL analysis. Thank you.
Very much. Let's raise hands for Kaur. There are 2 questions. Jason Torres asks, when you evaluate variant associations with a gene co-expression module, what value do you use for the module? Is it a linear combination of the expression levels for each constituent gene in the module, or is it,
it's basically a linear combination, but it slightly depends on the method. So if for ICA, So the linear combinations are weighted by how strongly the method, kind of the gene contributes to the module. So for IC and peer, it really, the linear combination is weighted by to how strong is the loading of this gene for this co-expression module. For WGCNA, it's basically just the mean expression of the genes in the cluster.
Yes, thanks. Veera Yaragopal asks, is it common practice to residualize the gene expression for covariates that often include first NPCAs or peer factors derived from the gene expression matrix to remove noise? However, including peer factors as covariates removes not only noise, but also the correlations across the genes. How did you handle this issue in your analysis?
That's an excellent question. So there's a very nice paper. We have an upcoming preprint, hopefully out next week, that describes these results. But there's a nice paper demonstrating that for trans-EQTLs, if you residualize the gene expressions, if you regress out PCs or peer factors, it's very likely to introduce collider bias. So you can actually get false positive associations if you residualize gene expression data for trans-EQTL analysis. And so what we've actually decided to do is we don't regress out these kind of principal components from the gene expression data, just to avoid this collider bias. It might reduce our power to some extent, but I think it's probably the most appropriate thing to do here.
Yeah, thank you. Thank you very much. We have to move on. I hand over to Jan.
Okay, thank you. Our next speaker is Xia Shen, speaking on high definition likelihood inference of genetic correlations across human complex traits.
Yes, just trying to find my slides. Do you see my slides?
Yes.
And okay. Hello, everyone. It's my pleasure to be here talking about my work at EMGM, and I would like to thank the organizers to give me this opportunity. So my work today is on a method called high definition likelihood, or HDL. That's not lipids, it's a method in this case, as a comparison to the popular ALT score regression method for estimating genetic correlation. Just a quick brief overview of the definition of heritability and genetic correlation. So they are both parameters based on the variance of the components of the phenotype, right? So if we model the phenotype as a random model with a G component for genetic effects and E for the residue or environmental components, the variance can be partitioned so that the proportion of genetic variance is defined as heritability, and the covariance standardized by the variance of the G term is going to be genetic correlation. So in order to estimate this, in the genomic era, with the data we have now for the genome-wide information, there are basically two lines of approaches, right? So you can estimate this using individual level data, where if we look at the linear model that the genetic effects and the genotype has a linear form combination for the phenotype, then through a random effect model or linear mixed model, we can get these parameters in the covariance matrix of the genetic effects. And for instance, in the software like GCTA, you can construct the genomic kinship matrix, or we call GRM. Eventually, you estimate these parameters through linear mixed models. While this is kind of a time-consuming thing to do with large sample size, it was really nice that we have the method of ALT score regression that can use GWAS summary statistics to do that, which especially give unbiased estimates or consistent estimates for the genetic correlations. The idea of ALT score regression, if you know, is actually quite smart. When we first look at it, the expectation of the chi-square statistic actually have a regression form, right? On the slope, we have the heritability parameter for the univariate setting, and for the bivariate setting, we have genetic covariance on the slope. So eventually, we can compute the genetic correlations through this. So I note here, I get the laser pointer out, that the expectation of the chi-square statistic is actually the variance of the Z statistic, and the expectation of the multiplication of two traits Z scores are actually the covariance. So if you look at the form in statistics, this is so-called something like a method of moment estimator, right? And it is known that we can usually improve the method of moment estimate using a likelihood approach. So in this case, so we need to consider the likelihood setup for the particular model, okay? So after some reasonably simple math, you can derive the variance covariance structure of the Z-scores as this form, okay? It looks quite like LD score regression. That's because if you define the LD correlation matrix R as R, so R squared or R prime R, because it's a symmetric matrix, we get an L matrix, where here I call it LD score matrix. So on the diagonal of this, you happen to have all the LD scores, okay? So that gives you the LD score regression, which is a method moment estimator. But off diagonal, these blue parts, they also have, they are non-zero numbers, right? So they have information also here. So if you consider all these, we can hopefully boost our power in terms of testing genetic correlation. So the way we do it is, of course, mathematically as straightforward as this. For two traits, both their heritabilities and genetic covariance are sitting in the variance covariance matrix of this block matrix setup. So mathematically, if you maximize this full likelihood, we can get all these parameters and the genetic correlation estimate as well. I'm going to skip all the technical part of this because there's a lot of numerical tricks we need to do in order to do this big model. But let's go to the results. So once we have this implemented, the method, so again, the HDL method here stands for high definition likelihood. It's not a lipid. The method actually has better efficiency as expected compared to LD score regression. So in a simulation where we set up the true genetic correlation to be 0.5, through a number of replicates, we can see that the efficiency gain is quite obvious. So it's as if you have double the sample size. So the variance of the estimates got reduced half. So there are two settings in the plot. One we call high heritability setting, so where the two traits are reasonably highly heritable. So the average heritability, if I remember, was around 0.7, and the other one is low group, where the average is about 0.3. So this looks quite good. So we apply this to a set of UK Biobank phenotypes. There are 30 in this plot. So 435 pairs that we need to estimate. So at the upper triangle, you have the estimate from LD score regression, and the lower triangle is the likelihood method. So if you look at the point estimate, right, the color look quite symmetric, meaning that both the methods give very consistent estimates. But I marked in the squares, there are the additional significant pairs or genetic correlation we can detect at certain threshold compared to the LD score regression. So the number is about 35% more. So for those of you, I have some colleagues interested in lifespan, for instance. So there's a mother's age at death as a proxy for lifespan. We can see that the new method tell you if you have high blood pressure, if you're depressed, or have cancer, use too much mobile phone, you're risk-taking, they're just going to reduce your lifespan genetically. Another thing we checked is that, so there's always this discussion between LD score regression and linear mixed model, of course. So the estimates are usually different. And there are different reasons, but here I just want to focus on if you incorporate more LD information, you do get closer estimates to the linear mixed model if you consider it as a gold standard in this particular case. So a better correlation with the mixed model setting. And the improvement in terms of this is more substantial for binary traits or when there's a binary trait involved in the pair of phenotypes. So for instance, here we have depression bipolar and diabetes BMI. Diabetes bipolar, they are all having quite different estimates from two methods, but for all of them, the likelihood method give you much closer estimates to the linear mixed model estimates. So that's basically all the results I have for today. So in summary, the take-home messages here are that if you incorporate full LD information in the modeling of summary statistics for genetic correlation, it basically doubles the estimation efficiency as if the sample size is doubled. And we can actually close the gap between square regression and the mixed model, or GCTA, if you are familiar with, which is also what we expected. I also have to know that the consistent estimation of heritability parameter is quite difficult. When we do genetic correlation, the bias in heritability and genetic covariance cancel out in a way so that it becomes consistent. But for heritability, it's getting closer, but it still remains further studies. So with this, I would like to thank all my coworkers at 3 different places. In this research, particularly for my colleagues, my fantastic PhD student, Zheng Ling at Karolinska Institute, and Professor Udi Parvitin, who did, especially Zheng, did all the hard work on this. Also the Wilson group in University of Edinburgh, and my group of Chinese students in Sun Yat-sen University, 3 of them actually presented today, Ran Ran, Zhijian, and Ting. I would like to thank all the audience for listening to them again here.
Okay,
so that's it for today. Thank you.
Sorry. Thank you, Xia. That was a great talk, nice and clear. We have one question here from an anonymous one. It says, we know that LDSC needs large sample to work accurately. How about HDL? What is the minimum sample to have accurate estimation?
I'm just going to read this again. Regarding HDL, minimum sample size. Oh yeah, good question. So as I presented, so mathematically, you can say if you believe 10,000 samples is enough for LD square regression, I would say 5,000 is enough for HDL because the efficiency is doubled, right? We had a recent trial of this on 3,000 people in a proteomics data, where you can, the autosomal heritability is quite sparse for proteomics. It's actually working reasonably well. So I would say with thousands of people for complex traits, polygenic traits, it's powerful enough. That's my
okay,
clue
thank
now. Yeah.
you. Sounds good. Just to keep to the schedule, I think, no more question. Let's, thanks Xia again. We'll go to, I guess after this, there'll be another session where you can answer more questions. And let's go to the next speaker. Next speaker is Simone Rubinacci, speaking on fast imputation of low coverage sequencing data from very large reference panels. Are you there, Simone?
Yeah. Do you see it?
Yep, there. I see you now. Yep.
Perfect. Thank you. Okay, let's go. So, hi everyone. I'm very excited to present our new genotype imputation method for low coverage sequencing data designed for very large reference panels. So, in the last 15 years, we assisted to several genetic studies, for example, in the case of GWAS, where data was generating using genotyping arrays. In this context, the focus was mainly on common variants. And at rare variants, and we use this to loss of power due to imputation. To study rare genetic variation, iCoverage would be the gold standard. Anyway, it's too expensive, especially because we are talking about very large sample sizes of hundreds of thousands of samples, even millions. So in this context, low coverage whole genome sequencing has been proposed as a cost-effective alternative. It has been shown that low coverage imputation increases the power of genome-wide association studies, and is also very attractive from an economical point of view. For example, 1x coverage is half of the price of a very dense SNP array. The problem with low coverage sequencing is that there are no efficient computational methods that are able to handle this data. And this limited a lot the application of low coverage in genetic studies. The main difference between SNP arrays and low coverage sequencing is in the pattern of missing information. So SNP arrays are usually designed for specific populations and take into account only common variants. This is not the case for low coverage sequencing, where the missingness is more driven by the sequencing of coverage. And we have information both of common and rare variant. In both the cases, we run genotype imputation to fill in the missingness of the data, the genotypes that are missing. In this context, genotype imputation methods are statistical methods that are able to borrow information from a very large reference panel of haplotypes and complete the sparse picture of the data. In the context of SNP arrays, we have very efficient methods for imputation that are able to scale on reference panels in the order of thousands to million haplotypes. However, this is not the case for low coverage, even if the methods are very similar. And so for this reason, this motivated us to create a new method for low coverage sequencing imputation that is actually able to leverage information from large reference panels. And we call our model GLIMS, and I'm going to explain what it is. So the starting point of our model is the sequencing reads of our low coverage sequencing dataset. From this, we compute the genotype likelihoods, which is a probabilistic representation of the genotypes that it takes into account uncertainty of the reads. The goal of the model is to refine these genotype likelihoods and provide very accurate genotype posterior probabilities that can be used, for example, in association studies. To do this, we actually sample, upload, we run, so the GLIMS algorithm runs in iterations, and for each iteration, at each individual, it generates a new haplotype estimate, so a new pair of haplotypes for each individual. To do this, GLIMS uses two main key features. One is the haplotype selection model or state reduction, where the states in our model are the haplotypes in the reference panel we are copying from. And we use a very efficient data structure called positional Burroughs-Wheeler transform that is able to subset the haplotypes and just take a very small subset of haplotypes for each target haplotype or individual. From this small subset of haplotypes, we can then do some haplotype sampling that in our case is slightly different from other models. Other models in low coverage sequencing scenario use diploid step, which is usually quadratic in the number of haplotypes or number of states. In our case, we do two different diploid steps, and these two steps are very quick and is actually linear in the size of the states. And we are able to generate two haplotypes for each individual. At the end of all the iterations, we will produce a set of genotype posteriors and also phase data. So I explain a bit what GLIMS is about, and one question is now how the GLIMS compares to other methods. So I have to explain a bit what imputation accuracy is, because it's not straightforward. So here I show imputation accuracy for 1x coverage. The data I'm showing here is just European samples that have been imputed, and the data was generated from 1000 Genome Project, New York genome samples at high quality at 30x, and we downsampled the reads to 1x. And then we used one as a validation, the 30x dataset, and the other one as a target to impute. Then imputation is typically measured, imputation accuracy, as the R-squared correlation between imputed and, in this case, our validation dataset, some high quality code. And we plot the R-squared correlation against the minor allele frequency. I'm not sure you're seeing here on the x-axis. In this case, I'm showing the results for BEAGLE 0.01, which is this 4.1, which is a standard model for low coverage sequencing. It's very used. It's the state-of-art model at the moment. And as a reference panel, we use the HRC reference panel. However, is BEAGLE 4.1, in the case of low coverage, is not able to leverage the size of the HRC. So we have to downsample to 5,000 individuals in the reference panel. And this is the result I'm going to show, just with a reference panel of 5,000 individuals. We can therefore run other methods for low coverage sequencing, such as GenImp and Stitch. GenImp does perform some approximation, and there is a slightly reduced accuracy, while Stitch is really not designed to take into account large reference panels. It's mainly for species without a reference panel. So there is a drop in the rare variation here. If we plot the accuracy of GLIMS, we see an increased accuracy, especially in a rare frequency variant. And this is due to our model, however, it's also possible to increase the size of the reference panel using GLIMS, but it's not possible with other methods. And we also obtain a boost in terms of accuracy due to the fact that we leverage information from a large reference panel. What is stunning here is that all these accuracy results that are obtained with a small fraction of the time of all other methods. And this is what I'm going to show here. So we run, this is the reference panel size in the x-axis. We downsampled the reference panel from 500 to 20,000 samples. And we ran an imputation experiment for each of the imputation methods. On the y-axis, I show the running time per sample, and it's actually on a log scale. So unfortunately, few configurations, especially for large reference panel sizes, were not able to run in time. For these reasons, we extrapolated the data from what we had. We can see that all the other methods are scaled quadratically or linearly in the size of the reference panel sizes. And the stunning feature of GLIMS is that actually the running time decreases as the reference panel size increases. And there is also an increase, like the difference between reference panel 20,000 between GLIMS and BEAGLE 4.1 is approximately 3 orders of magnitude. So another question we can ask is how imputation is affected by changing the coverage, and how we do compare against SNP arrays. For these reasons, we generated 38 different configurations, changing the coverage and also simulating other 25 different SNP arrays. And we made all the results available on our website. So in this case, we show 503 European samples as before, but in this case, it's not chromosome 1 data, it's whole genome. And we can see that imputation accuracy increases by increasing the sequencing coverage. This is, of course, expected because the amount of missing data is decreasing by increasing the coverage. In addition to this, we can compare 1x coverage, for example, to well-known SNP arrays. For example, the Illumina GSA array, which is a sparse SNP array, and the Illumina Omni 2.5 array, which is a very dense SNP array. in this case, we see that at common variants, approximately we have the same imputation accuracy. However, 1x coverage is much better at rare variant. And this is because, again, 1x coverage has much more information at rare variants compared to SNP arrays. Another interesting question we can ask here is, since SNP arrays are actually biased towards some population that has been involved in the design of the SNP array itself, what happens if we measure imputation performance using SNP array and low coverage in the case of different populations, not well represented on SNP arrays? This is what we did here. So we have imputation performance on 61 African American samples. So this is our 1x coverage. And we can see how the imputation performance changes. And this is due to the fact that the SNP arrays are not designed for this population. Well, this bias is not present in low coverage. And this is also true for the Illumina Omni 2.5. It's a very dense study. So finally, we saw these results and we asked ourselves how this increased accuracy could translate into downstream analysis. So we use all these 38 calls in order to run to map eQTLs using JWAD's expression data. This is what we do here. So on the y-axis are all these genotype calls we generated before. And then on the x-axis, there is the fraction of genes that for which the expression is significantly associated with a variant. And we can see that even a low density, low coverage like 0.5x is actually getting very similar results with very dense SNP arrays. And 1x is outperforming all other SNP arrays. Similarly, motivated by the fact that low coverage outperforms SNP arrays, especially the rare variants, we wanted to run, we ran a burden test analysis. In this case, we compute the correlation between the imputed burden, so the rare variants overlapping exons, and 30x data considered as the ground truth. In this case, we see that 1x has approximately the same correlation, the same performance, as the Illumina Omni 5 exon, which is a SNP that is the most dense SNP available on the market, and it's also being enriched by exon regions. So overall, these analyses show us that low coverage imputation can be used for functional analysis, and this has been reported already in previous studies, and we confirm this. And I would like to conclude with few remarks. So GLIMPSE is a method for genotype imputation. It is part of a new generation of methods that are able to efficiently leverage reference panel information from very large reference panels. We show also that 1x coverage provides very accurate genotypes, and while being very economically attractive. And these results can potentially have a big impact in association studies, but also in population genetics, and also ancient DNA, for example, where low coverage is more used. Finally, I would like to thank people from my group at the University of Lausanne. And also, for those of you that are curious, we have just yesterday released a preprint of our paper, and it would be useful to get feedbacks. Thank you very much.
Thank you, Simone. That was a very clear talk. Every time I thought I had a question, you answered it on your next slide. But I see there's a couple of questions coming up here now. Sure. One from Nicola Parastu. How much do you think you would gain in accuracy using the TopMed imputation panel?
Sure. We will have, yes, it's a very good question. The TopMed imputation panel is revolutionizing the reference panel. So we will get a clear boost at rare variants, definitely. We also will have much more minor allele frequency to look at. So with because it's high coverage sequencing. So it's a new generation of reference panels. We'll get a clear boost compared to other methods. There is some results in literature about what's the gain in SNP arrays, and I think it's probably the same in terms of what we gain. Like, yeah.
Okay. That answers it. Thanks. And the next one from Irini Maruli. Great talk. How does GLIMPSE compare to IMPUTE-2?
Yeah, IMPUTE-2, there is actually a new method called IMPUTE-5. I'm an author of them. I'm the author of IMPUTE-5. And approximately, I think I changed slightly the question. One interesting question is how does low coverage sequencing imputation compared to SNP array imputation in terms of performance? This is something I didn't talk about. And we show in our paper that it's approximately 1 order of magnitude with the best available methods, which is IMPUTE-5 and BEAGLE 5.1. So there is still a cost. However, this cost is absorbed in the fact that low coverage sequencing is not so much cheaper compared to SNP arrays, or at least at the same price. And so we can do whole genome imputation using the HRC for approximately slightly less than $1. So I think this is just a small fraction of the cost of an analysis. Hope I have answered correctly.
Oh, that's fine. Okay, thank you. And that's the end of the session.